Good morning. Myself Rosita D'Souza, and I'm working under the guidance of Professor Rizwan Sheikh. The topic is crop disease detection using machine learning. We know India is an agrarian country and its economy is largely based upon the crop production. Agriculture backbone of every economy, and India, which is ever increasing demand of food due to rising population, advances in agriculture sectors that requires the need of the person. The agriculture sector needs a huge upgradation in order to survive the changing condition of every economy. To get the optimum yield, the crop should be healthy. Therefore, a high technical method is needed for periodic monitoring of the crop. Crop disease is a major factor which indirectly influences the reduction of both quality and quantity of agriculture product. There are number of pesticides to control the diseases and to increase the production of the crop, but to find the current disease and to later find a pesticide to control the affected disease is quite expensive and time consuming. The presence of disease on the plant is mainly reflected by the symptoms on the leaves. So there is a need of an automatic, accurate and a less expensive machine vision system for detection of diseases from the image and suggest a proper pesticides as a solution. Plant diseases. There are two kinds of plant diseases, a wheat plant disease and a cotton leaf disease. The diseases which affects wheat plants are leaf rust, powdery mildew, stem rust and yellow rust. And diseases which affect the cotton leaf diseases are bacterial blight, alternaria, cerospora, grey mildew and fusarium wilt. So let's see the diseases which affects the wheat plants. Leaf rust causes a small brown lesions on the leaves. These blister-like lesions are common on the leaves and occurs on the leaf sheath, which extends from the base of the leaf blade to the stem node. These lesions are normally smaller, more round, and causes a less tearing of tissues than caused by the stem rust. Next is powdery mildew. As we see in the image, a powdery mildew causes a white lesions on leaves and leaf sheaths. And glumes and awns are also affected when the disease is severe. The fungal growth is largely limited to the outer plant surfaces and can be easily wiped out by rubbing a finger across the affected area of the leaves. The mature lesions have a dark reproductive structure mixed with white cottony growth of fungus. Stem rust. A stem rust causes a blister-like lesions on the leaves, leaf sheaths and stems. As same as the leaf rust, glumes and awns are also affected. The reddish-brown spores of the fungus causes a tearing as they burst through the outer layer of the plant tissues. Matured stem lesions are more elongated than those of leaf rust. The next is a yellow rust. Yellow rust is usually known as a stripe rust, which is a fungal disease, and it produces a leaf lesions which are yellow in color and they are grouped into a patches, and it is of a 2 to 3 millimeters wide that run parallel that run parallel to the leaf veins. So yellow rust is responsible for approximately 73 to 85% of recorded yield losses, and grain quality is also significantly reduced. So the next is the cotton leaf diseases. The first is a bacterial blight. Bacterial blight is a bacterial disease mainly caused by a bacteria Xanthomonas campestris malvacearum. So the symptoms are a bacterial blight has a starts as a dark green water-soaked angular spots of 1 to 5 millimeters on the leaf with a red to brown border. At the beginning, these leaf spots appear as a water-soaked area, which in later on changes from a dark brown to black color. The spots on the lesion areas of leaves may spread over the major veins of the leaf, and in later petioles and stems get infected and the fall of leaf occurs. Alternaria. It is a fungal disease which is caused by A. alternata or Alternaria macrospora. The symptoms are at the beginning, um, it is brown or grey brown to tan color. A small spots appear on the leaves which varies from 1 to 10 millimeters in size and which later on becomes a dry dead leaves with a grey centers and a fallout occurs. Sometimes old spots combines together and creates irregular dead areas on the leaves. Cerospora. It is a fungal disease which is caused by a Cercospora gossipina. The leaves have a red blemishes on the leaves which expands the distance of around 2 centimeters. The spots are sometimes round or irregular in shape with yellowish, purple, dark brown or blackish fringes on the leaves. This shows up because of the limitations of lesion region by veins of the leaf and influences a more seasoned leaves of a well-developed plants. Grey mildew. It is a fungal disease and caused by Rumeria areola in imperfect stage and Microsporella areola in perfect stage. At initial stage, infection appears as a triangular, square, or irregular circular whitish spots of 3 to 4 millimeters size on the leaves. These diseases primarily appears as irregular angular and sometimes a pale translucent spots of diameters 1 to 10 millimeter on the older leaves of the matured plants. The infected regions are light to yellowish green in color on the upper surface. As disease infection increases, spots merge together, forming a bigger spot, and the leaf tissues turn yellowish brown, while whitish frosty growth appears on the lower surface of the leaf, but occasionally on the upper surface of the leaf. Fusarium wilt. It is also a fungal disease caused by a Fusarium oxysporum. Most the diseases are showed when the plants are grown up and are more mature. The plants first get a distinctly a darker green color and laterly turns to the yellowing of the leaves and the loss of foliage is observed. At first, it shows up on the lower leaves around the season, and then leaf edges shrink and turn yellow initially, followed by brown with the inward movement on the leaves. Proposed system. Proposed system provides a solution for regularly monitoring of the cultivated areas and provides the automated disease detection using remote sensing images. So there is an objective of the proposed system is to early detection of diseases as soon as it starts spreading on the outer layer of the leaves. The proposed system has two phases. The first phase deals with training dataset, which includes a training of both healthy and diseased datasets. The second phase deals with monitoring the crop and identifying the diseases using Canny's edge detection algorithm. So once the dataset is ready with healthy and infected image samples, the threshold is extracted for both aging and for diseases in a training dataset. Periodically, images are obtained by a remote sensing. So RGB values of the monitored images are extracted and compared with threshold images. If the threshold is greater or less than given value, then histogram analysis and edge detection techniques are used to identify the particular plant diseases and to find a particular solution for that diseases. A lot of healthy and defected crop images are considered, and for each crop a threshold value has to be set. The model should be trained in such a way that it should take a proper decision for all the crops. So different types of crop images are given as an input to the training model. Training model consists of a healthy crop images, infected crop images, and different disease crops, and even the pesticides for that diseases. And different resolution images are also obtained from the agricultural satellites. A disease detection. We are taking a reference image as an input to the decision support system where we are using a decision algorithm to identify the different kinds of diseases. This system makes use of a trained model to take the further decision. The trained model uses the dataset to provide a proper suggestion. If there is a huge change in threshold of the reference image, then check whether it's due to aging. If it's due to aging, then no need of imitating it. But if it's due to diseases, that is deformities of leaves and color changing in the leaves, then it is identified and edge detection and histogram analysis will be carried out to find a proper solution and a pest as a pesticides for the particular disease. Canny edge detection algorithm. Canny edge was developed by John F. Canny in 1986. Canny edge detector is an edge detection operator that uses a multi-stage algorithm to detect a wide range of edges in image. A multi-step algorithm that can detect edges with noise suppressed at the same time and smooth the image with Gaussian filter to reduce noise and unwanted details. So the process of Canny edge contains 5 steps: noise reduction, gradient calculation, non-maximum suppression, double threshold, and a track edge by hysteresis. So a one way to get rid of noise on the image is to filter the image with derivatives of Gaussian, that is Gaussian filters. The second step is a gradient calculation. Gradient calculation step detects the edge intensity and directions by calculating the gradient of image using the edge detection operator. And the non-maximum suppression. The final image should have thin edges that we perform non-maximum suppression to thin out the edge. The fourth is double threshold. Double threshold has three kinds of pixels: strong, weak, and non relevant. So, strong pixels are the pixels that have high intensity. Weak pixels are the pixels that have intensity value that is not enough to be considered as a strong one. So, other pixels are considered as a non relevant for the edges. In threshold, there are two kinds of: the high threshold and low threshold. High threshold is used to identify strong pixels, whereas low threshold is used to identify the non-relevant pixels. The last step is the track edge by hysteresis. This finalized detection of edges by suppressing all the other edges that are weak and not connected to strong edges. Based on the threshold results, the hysteresis consists of transforming weak pixels into strong ones, if and only if at least one of the pixels around the one being processed is a strong one. So weak edges that are connected to the strong edge will be removed. To speed up this process, algorithm keeps the track of the weak and strong edges, and that way it can recursively iterate through every pixel of image. Finally, we iterate through the remaining weak edges and set them 0, resulting in the final processed image. This is the algorithm for disease detection. Then read the reference image, that is either a crop image or a leaves image. Then check the threshold. If change in threshold is due to aging, then it doesn't matter, so directly stop. Else convert the image into grayscale, then apply a canny edge detection algorithm and get the histogram value. Once everything is done, identify the particular disease for the reference image. So the first step starts the detection of plant diseases. RGB images of healthy and infected plants are given. First, we give two samples, one for the healthy images and second for diseased image. In healthy image sample, we choose a normal or uninfected images of leaf. On the other hand, we choose the infected image of diseases. The detection of a plant diseases starts with a training process. In training process, the edge detection technique cannot apply directly on the images. First, we need to convert the image into grayscale. Then, Canny's edge detection technique is applied. First, separate the layers of images into red, green, and blue layers. Then, apply the Canny's edge detection technique to detect the edges of layered images. This technique is applied on both the samples, such as the healthy sample as well as disease samples of same kinds of plants. So choose the test sample of plant. When the testing sample is selected, the training process is started again on the testing image. So in the training process, first separate the layers of tested image into red, green, and blue layers and again apply Canny's edge detection technique to detect the layers of the images. Once training process of the samples is finished, histogram is generated for both healthy leaf sample and diseased leaf samples and saves in the memory. These histograms are displayed when we generate the histogram for the testing images. In the second phase, after the training process, the histogram testing sample is created or generated suddenly. Once the histograms are generated for both samples and testing images, we'll apply the comparison technique based on the histogram and edge detection technique. The comparison is firstly with testing sample and healthy sample. If the testing sample is diseased, it compares testing sample with diseased sample, and this step takes to display the comparison results, that is testing sample is diseased or not. Our system monitors the cultivated fields, and crop diseases are detected in early stages by using the edge detection and histogram matching. And machine learning techniques are used to train the model, which helps to take the proper decision regarding the diseases. The pesticides are suggested as a remedy to the farmers for the infected diseases and to control and to um, and to cure the diseases by using a proper pesticides. It is efficient and accurate technique for edge detecting an image significantly and reduces the amount of data and filters which are useless. So while preserving the important structural properties in an image, also the healthy and infected samples of images are saved into database for further training process. So in future the proposed system are implemented by adding extra services like providing the pesticides with the price leads to the farmers so they can decide the proper pesticides for their crops and to overcome the diseases. Thank you.